Hello everyone and welcome to my channel. Let's begin with the topic immune system organs. So we have primary organ and secondary organs of the immune system. The primary organs include bone marrow and thymus. So bone marrow is basically a site for immune cell production as well as it helps in B cell maturation, and thymus is the place where T cells mature. The secondary organs include spleen, lymph nodes, tonsils, Peyer's patches. Okay, so what does these help? They help in interaction of the immune cells with the antigen. Let's first begin with lymph nodes. So, lymph node is basically a secondary lymphoid organ. It has many efferent and one efferent lymphatic vessel. So these are many afferent ones, and this single one is the efferent lymphatic vessel. This whole structure, it consists of a capsule, and there are present some trabeculae, which are the indenting spaces. Okay, and these functions are basically, they help in filtration of macrophages, B and T cells. So basically your lymph is going through these lymph nodes, right? Now these lymph nodes, it consists of follicles, that is this, we have primary and secondary, these are the follicles. Then we have medulla. So basically there is a cortex. Cortex consists of follicles. Then we have a paracortex, which is this pinkish region, and then this purple region is your medulla. Okay, so what does this follicle do? So basically follicle is present in the outer cortex, and it is the site of B cell localization, as well as B cell gets proliferated here. So these follicles, they are again primary and secondary. The primary ones, they are dense. See, this primary follicle, it doesn't have any, like central, you can say, pale area. It is all dense, and it is quiescent. It is inactive, you can say, right? Whereas the secondary one, the secondary follicles, it has a germinal center which is pale. The bluish one is basically the germinal center, and these are the ones which are active follicles. Then comes medulla. Medulla, it consists of, we have medullary cord as well as medullary sinuses. So medullary cord is this area, number 2, you see the purple ones inside that, and then here we have the sinuses. Okay, so what are the functions of these medullary cord? Medulla consists of, as we just said, we have cords and sinuses. The cords, basically they are concerned with plasma cells and some of the lymphocytes, whereas sinuses, that is medullary sinuses, they consist of they are more concerned with the macrophages. That means these in your lymph nodes, where is the site to find out macrophages? That is in the sinus, that is medullary sinus. It is the site where macrophages will be present. Medullary cords is the place where plasma cells are present. Okay, then what we have lastly we have paracortex. Now this paracortex is the area where we have the T cells. So we are done with B cells, we are done with macrophages, now comes the T cells. So paracortex is the area where T cells are present. This is basically the region, as we just said, it is present between cortex and the medulla. And what happens is that via these afferent vessels, venules basically, the B and T cells, they enter from the blood and they come and get collected, those T cells get collected in this paracortical area. Now, applied to this is Dye-George syndrome. So, in case of a Dye-George syndrome, there is basically underdevelopment of this paracortex area. It is basically a defect of third and fourth pharyngeal arches, and along with the paracortex, your parathyroid hormone is also affected in this condition. Another thing to remember is that in cases of lymphadenopathies, so it is this area which gets enlarged, the T cell containing area, that is the paracortical area, in response to the other immune system. So just to summarize this, so from the site of the infection, the lymph, it will drain those antigens, okay, and those antigens will be drained out through the lymph, and it will come via these afferent vessels and enter into the lymph node. Then there will be present some antigen presenting cells like dendritic cells. They will present it to the T cells or the B cells as required, and then they will further process these antigens. Will process and they will present it to these immune cells, and then further immune reaction will take place. Okay. Some interesting points about these lymph nodes to remember is that the right lymphatic duct, it will drain the right lymphatic duct will drain the right side of the body, that is above the diaphragm. All the lymphatic structure from the right hand corner will be draining into right subclavian and then into the internal jugular vein, whereas in the left side, the thoracic duct which is present, the diaphragm which is present, it drains below the diaphragm, and the left thoracic, the upper limbs, they are going to drain into the left subclavian and the internal jugular vein. Okay, so this is about the lymph nodes. The second structure is spleen. So, spleen is basically an organ which is present in your left upper quadrant, and it is present in relation to your left kidney. It is present anterior lateral to that. Now, spleen, it consists of a capsule, then there are again some trabecules present. Here we have a red pulp and a white pulp. The red pulp, it is basically concerned with RBCs, and white pulp with WBCs. Let's first study about this red pulp. So a red pulp, it contains sinuses, that is a red pulp consists of sinusoidal spaces as well as some fibrous network. From this red pulp, there is a drainage to these veins and arteries going on from this red pulp. Okay. Now, then we have white pulp. In the structure of white pulp, we have just outside this this white pulp area, we have PAL, that is periarteriolar lymphoid sheath. Okay, so this is the structure which is concerned with T cells. Then we have the marginal zone. Marginal zone is concerned with macrophages. After that, we have the main follicle area, which consists of a germinal center and a mantle zone. So this will be the mantle area of the follicle, and then the core central area will be the germinal center. So basically these both consist of structures of follicle. So if I may repeat, we have T cell area is present periarteriolar sheath. Then we have marginal zone, which consists of macrophages, and then the follicle area, which consists of B cells. Marginal zone basically is the area where antigen presenting cells will present their antigens to the macrophages. Okay, and where is it present? It is present basically just at the just between the red and the white bulb, as we just said, just the corner ones only, the junction, you can say. Okay, then if we talk about certain applied aspects of spleen, so see, whenever there is splenectomy done, so why do we do splenectomies? It could be because of trauma, or patient is suffering from some conditions like ITP, or like hereditary spherocytosis and these conditions. In those, we do splenectomy. So whenever we do splenectomy in patients, they are more predisposed to infections by encapsulated organisms. Like most common one is pneumococci, and therefore you need to vaccinate these patients. So other ones include H. influenzae and meningococcus as well. So you need to do these. Also, there are certain microscopic findings like Howell-Jolly bodies. Basically, these are the nuclear remnants. So you know that old dead RBCs, they are brought to the spleen, and they are broken down. So when spleen is not there, so those nuclear remnants of the will be kept on there only. Then comes target cells. Target cells are the cells, you see, basically just like a target, on when you'll see under microscope, the cells will be in these shapes. So these target cells are again often found in after the splenectomy patients. They can also be found in liver diseases and patients having renal diseases and the post splenectomy. So whenever there is spleen dysfunction, what will happen? These antigen presenting cells won't be able to function well. Okay, there will be a decrease in the IgM. So there will be decrease in IgM as well as IgG immunoglobulins. There is decrease in the activation of complement system. Complement pathways won't be activated. There will be no, you can say, opsonization won't occur. There will be increased susceptibility to infections. The ones just we talked about, that is these encapsulated organisms, and the infection will increase. Okay, now next comes thymus. Thymus is another important organ. It is located in your mediastinum. To be exact, it is present in your anterior mediastinum, anterosuperior, and T for thymus and T for T cell. So basically thymus is concerned with T cell differentiation as well as its maturation. This is again encapsulated. Every other, just like other lymphoid tissue, these are also encapsulated ones. And in this thymus we have a cortex and a medulla. Some other important points are that T cell, this thymus epithelium, it is basically derived from third pharyngeal arch. That is, it is endoderm in origin. Another structure which we just did was which is derived from third pharyngeal arch is the parathyroid area of the lymphoid follicles, right? Lymph nodes. So that is also from the third pharyngeal arch. Whereas the cells itself, the thymic lymphocytes, they are mesodermal in origin. Okay, coming back to cortex and medulla, basically cortex, cortex is the area where there is immature T cells present, and medulla is the area where they will be matured out. So how does this whole process occurs? So basically there in your thymus there occurs some positive and negative selections so that we can rule out what cells are to be discarded out. Okay, so in cortex there occurs what is known as positive selection. In this, the thymus epithelial cells which we just talked about, they will express the major histocompatibility complexes. Now T cells are tested that how much is their affinity for binding to the self MHCs. If there is weak binding, matlab that they are not able to bind very properly to these MHC, that means their function, they are not the strong cells, T cells. So those cells, if there is poor binding, those cells will be apoptosed. Similarly, in medulla, we have a thing of negative selection. Here, the epithelial cells, they will express self antigen, and so T cells, they are tested against these self antigens. If there is too much binding, excessive binding to these self antigens, which it is of course harmful, so these cells are again they undergo apoptosis. Otherwise there will be increased risk of autoimmune diseases. Okay, now next applied is regarding, as we just discussed about Dye-George syndrome, right? So Dye-George syndrome, which is a defect of third as well as fourth pharyngeal arch. So in this, first we just discussed that parathyroid area of lymph node, it is not there. Now we discussed that thymus. So the thymic shadow, which is usually present in children, they will be absent here, and even the parathyroid gland is also absent. So the patient complains of hypocalcemia. And then there is a tumor, that is thymoma. So thymoma is basically a neoplasm of thymus, and it is associated with various like superior vena cava syndrome, then in patients with myasthenia gravis or pure red cell aplasia. So these are the conditions which are associated with thymoma. Another interesting thing is that, see, we talked about this thymic shadows now. So till what age this thymic shadow is usually present on chest X-ray? Till 3 years. After 3 years, it will involute. And how is this? It is basically a sail shape shadow. Thank you very much for listening. Let's keep on learning together.